Yu-Ching, thank you very much for inviting me to your uh, meeting here. Also, as you said, it's a little late for me or too early. But since Corona, this happens very often. I'm on this side on the earth. I'm always either at morning at 6 o'clock or at midnight, something like this. And yeah. Next to the talk you asked me to give about chemical fixation, I feel a little bit strange in this because during my PhD, I was not allowed to do chemical fixation. In Martin Müller's lab, this was a no-go. Everything was based on cryo-fixation and freeze-substitution. And I didn't know anything about cryofixation until the end of my thesis, when I had to give reasoning why I was using cryofixation, free substitution. Then I started to read a lot of papers, and now I speak more about chemical fixation than about what I really like, cryofixation and free substitution. Anyhow, today I will also have a few words about that. Then let's start. What do I do? Yes. Okay, so we have the different preparation steps like the chemical fixation and then dehydration, and then we would have resin embedding. And I added cryofixation in here because, as I said, I will also spend a few words on cryofixation. Now, why do we do a chemical fixation? We need to protect the ultrastructure from collapse and loss of cellular components because, as everybody knows, we cannot really, now it's getting maybe a bit difficult to stay on that statement because there are exceptions as everywhere, but we are not really able to look at our samples in its watery surrounding. So we need to remove the water. And for this, we need to use organic solvents. And with this, we would also lose a lot of cellular components. So that's why we start with a chemical fixation. One that cross-links proteins. These are aldehydes. And then we have cross-linking of lipids, which is done with uranyl and ions and osmium tetroxide. And of course, as you will see, I will show a few things. This can lead to distortion of protein, can lead to proteolysis. And if you want to do immunolabeling, it also has a negative effect on the epitopes. And one of the most well-known aldehydes is formaldehyde. And this is how formaldehyde really looks like. It's a polymer. It's quite a long polymer. And if you look on this here, then you will see that it is a powder. So this is one of my fights as Don Quixote, namely to tell that nobody of you ever was using paraformaldehyde for doing chemical fixation. Paraformaldehyde cannot be used because it's a powder, and everybody who writes they use paraformaldehyde in their papers, in fact, is lying. Because you depolymerize it, usually with some temperature and hydroxide, sodium or potassium, usually sodium, and then you get the molecule you're working with, then you get formaldehyde, and this will work as a fixative. If you add it in water, it's more specifically methanediol, and this methanediol, as you see here, can then add to amino groups and cross-link proteins. And cross-link proteins. And as you see here with the two arrows, this is quite important to remark. This reaction is reversible. So if you remove formaldehyde, let's say if you wash your sample afterwards in a phosphate buffer, let's say, and leave it over ages, then formaldehyde will detach and the fixation will be inverted. And maybe also to mention here, before it's too late, formaldehyde in fact is a gas that you dissolve in water. So that's also when you smell above the vial, you smell the formaldehyde gas. So this is also, let's say, the formaldehyde goes then, if you have sample in phosphate buffer, formaldehyde will, or the fixation will be reversed. You have formaldehyde then in the water, and this will then, or can evaporate. So at the end, you have no fixation anymore. For this, glutaraldehyde is much better fixative. It also reacts with amino groups. It cross-links proteins. It's a big molecule, as I will show you a little later, so it is slow in penetrating. That's why I very often use formaldehyde and glutaraldehyde together as a fixative. So let's say formaldehyde then to be the faster going into the cell, starting to fix, and then glutaraldehyde following thereafter and how do you say, stabilizes the fixation. And the cross-linking is irreversible, and you have to take care that you have good buffers because you get pH drop. And it can have big influence on antigenicity if you want to do immunolabeling. So now how does the molecule look like? So this molecule, this is glutaraldehyde, as you can buy it in its pure form. And this would react with amino group like the formaldehyde did in a similar way, and building this Schiff base here. But this reaction is reversible, and this is glutaraldehyde at low pH. But as you probably all do, you work at pHs, let's say 7, 7.2, 7.4. And at that pH, glutaraldehyde polymerizes actually in a dimer, but I have read papers that it can also be a longer polymer than only a dimer. But now, when you look at this situation, then you see that you have the aldehyde group with a double bond to the CH group. Then you have a single bond, and again you will have a double bond. And this makes the reaction so stable because the electrons now don't know where they belong to anymore. The electrons are somehow going up and down or are distributed over this double bond, single bond, double bond. And it's called in chemistry, it's called mesomeric system. And with this, the fixation becomes stable or is stabilized because of this distribution of the electrons. And then maybe one of the artifacts, so one of my colleagues at the time when I did my PhD, Hans-Rudi Ebersold, he was working in our group for the microbiology department of the ETH. And he was looking at bacteria, as you see them over here. And as I said, we were not allowed to use chemical fixation in our lab. So he did the cryo-fixation with a propane jet and afterwards freeze-substitution. And as you see, he always got these very nicely laid membranes around the cell wall and also here at the septum. And so he got complications with his colleagues at microbiology because in all the papers you saw these structures, these vesicular structures here, these so-called mesosome at the time. And then he did the crucial experiment and checked. So he did chemical fixation and then cryo-fixation and freeze substitutions. That's what we see on the left. And what we saw before on the right was just cryo-fixation and freeze-substitution. And with this, he could prove that those mesosomes are an artifact introduced by the chemical fixation. So as you can see here, membranes can be destabilized by the fixation, probably through osmotic effects. Another example we did then when I was at the University in Utrecht, actually it was a PhD student of the medical faculty, Jean-Luc Murc, with whom we did the same experiments but with dendritic cells. And there was, let's say here, glutaraldehyde prefixation, and then the cells were high pressure frozen, freeze substituted, and this was without glutaraldehyde prefixation. And again, you see that the membranous structure somehow changed their morphology. So this probably should keep in mind when we think about sample preparation. Another things about glutaraldehyde is that it has influence on antigenicity if you want to do immunolabeling. And what this graph actually it should tell you is there is an influence, but the influence is antigen antibody or even epitope antibody dependent. So you cannot say if you use 2% glutaraldehyde, you don't get any immunolabeling anymore, as you would probably expect here for the DNase example. But it might still be working as you see it for the rabbit IgA example. So with this, we should consider that in everything what we are talking about in electron microscopy, we probably can more talk about tendencies and not absolute yes or no answers. I will show another example later on. Then we come to osmium tetroxide. Osmium tetroxide, actually here in this depiction, it already has reacted to CC double bonds here, or let's say of unsaturated fatty acids, for instance, lipids and so on. And with this, it can cross-link fatty acids among each other. So it reacts with CC bonds, can to maybe cross-link to proteins, but definitely to unsaturated fatty acids. As I will show in the next slides, it is proteolytic. It's quite fast in penetration, and most likely because of the proteolytic effect, it has a great influence on antigenicity. So this you see here, this was a colleague of mine at the Utrecht University who was using a Tanaka method, how it was called. So this was a professor at Totoro University in Japan, who was a specialist in scanning microscopy, and he wanted to use the scanning microscope to look into cells. So what he did was freezing his samples in liquid nitrogen, then taking a hammer and smashes it into pieces, and then collecting those pieces and put them in osmic acid. So he made several changes of osmic acids, and this is what my colleague did over here. And then you see now you can also look into the cells. I mean, the cells are broken open by the hammer, but you see most of the cytoplasm here has disappeared, whereas here the doliprane cap, which connects the two cells in this filamentous fungus, mitochondria with the cristae, multivesicular bodies with the vesicles inside, and so forth, vacuolar membrane, nuclear membrane, you can easily see, but cytoplasm has disappeared because it was chopped down and washed away during all these washing steps. You can also use it for, let's say, more artistic depiction of a cell. So this is another group from Koga at, I think he's at Hokkaido University, and he was using Tokia's cryosections, mounted them on a grid, and then treated them with osmic acid the same way as Tanaka proposed. And with this, again, he removed the cytoplasm, but kept membranes like the Golgi membrane over here, or the mitochondria, or endoplasmic reticulum, and so on. So just for me, these pictures are the demonstration that osmium tetroxide has a proteolytic effect. There are also papers on it, yeah, which support these findings. Then we come to uranyl acetate, which Ruching already said in some labs you're not allowed to use it anymore, which is in fact a pity because uranyl acetate binds to phosphate groups, so it can fix nucleic acids. It binds better to RNA than to DNA, and it can also fix phospholipids. Actually, it's the only fixative that I know that fixes nucleic acids. And here is just a tendency of immunoreactivity after different fixation protocols. Maybe the main important message from this slide you should get is that if you reduce the concentration of fixatives, in this case it's glutaraldehyde, then you get a higher immune reactivity. And in red here, these were two monoclonal antibodies, one against the protein moiety of this protein and the other one against the sugar chain. And then you see that the sugar chain, especially in case of osmium treatment, is more resistant or easier to label with antibodies. And this can also be shown with label like biotin antibodies against biotin and so on. So if you have a non-proteinous structure, if you have a sugar or another structure like that, then they are less influenced by the fixative than the protein. Now we come to the buffers, and here probably the buffers like HIPs, which use in cell cultures, are very good buffers. We mostly use phosphate buffer, and what I would like to fight again is forget about cocodylate. Cocodylate does not buffer where you need it to have it as a buffer. And I was going on search for how did cocodylate come into electron microscopy, but I didn't find any paper because I wanted to know what was the reason to use it for electron microscopy. I will come back to it. I just want to mention as last, if you want to use tris, which is also belongs to these good buffers, tris you can also not use, and I will show in a coming slide. So this is now the paper I found about cocodylate, and this is using cocodylate as a buffer because Blumel wanted to study the reaction of phosphatases, and then of course he couldn't have a phosphate buffer, so he needed to have another one, and he found cocodylate. And here what I'm saying, cocodylate doesn't buffer where you want it to buffer because the pK is about 6.3. And you should use a buffer in the range of 0.2, 0.3, maybe 0.5 pH scales above the pK. And you see also here, this is the titration curve, and a buffer works around this line more or less. but pH 7 is at the end where it doesn't work anymore. And some people even use it at 7.4. So I strongly suggest to change calculate. And why not using phosphate buffer? I didn't find a big reason why you shouldn't use phosphate buffer, and we use it regularly. And here is why tris is not useful, because tris has an amino group which would react with the aldehydes. And you can do the experiment. You can use tris and put glutaraldehyde on it. The solution gets yellowish. And in fact, what you're doing, you destroy the buffer capacity as well as the fixative. So don't use tris. It's very nice if you use it for immunolabeling, if you want to have a little higher pH to have a better antibody antigen binding, then it's very nice, but it's not, you cannot use it for fixation with aldehydes. And maybe one word I almost forgot about the buffers. There is maybe still, but at least in my time, there was a long discussion what kind of osmolarity you should use for buffers. And at that time, people were suggesting you should have about 300 milliosmol. That seems to be the osmolarity you have in blood. But there were many controversial papers. Some papers said it doesn't matter what molarity your buffer has. Some say the whole buffer and fixative molarity counts. But here imagine that if you add, I think, 2 or 3% formaldehyde, you already are above 1000 milliosmol and not by the 300 milliosmol. And then again, other people found out that from the fixative, there is only part really osmotic active. And maybe you're also working in a lab where you don't even have an osmolarity meter. We had one at ETH in Zurich, and since then I didn't see any anymore. So in my opinion, is don't really care too much about osmolarity. Also, because the different organelles in your cell will have different osmolarities. So, in my opinion, you can anyhow not get it right for every part in your cell. So now we come then to the dehydration, and dehydration we need because we want to infiltrate or we want to embed our samples in resin, and all resin we are using are more or less apolar. They are the Lovicryls and other metacrylates who can tolerate a little bit of water, but they are all on the apolar side. And we can do this in a graded series of ethanol or acetone. And they also, of course, have their effect for denaturing proteins. So, for instance, called acetone is used to precipitate proteins from protein solutions in biochemistry to concentrate the proteins. So you denature them, and with that they precipitate. But also you can dissolve lipids, of course. And here may be also another word. I also banned propylene oxide from my lab because propylene oxide is also very potent solvent, and you lose quite a lot of structure. So many people like those samples because they give you a very crispy picture. But this crispy picture, of course, is because you lose components. Then during dehydration also you will have shrinkage of the sample. So these were experiments done by Alan Boyd on mouse limbs who was studying dehydration effect. And what he found is in the beginning of the dehydration you get an increase of the volume. It's at about 70% dehydration. It's back to 100, and then it starts to shrink quite considerably. And here maybe even if you do a chemical fixation, using freeze-substitution as a dehydration might reduce this shrinkage. That's what we tried to show. This also from Boyd, who could show that if you use freeze drying, that the shrinkage was reduced. So now then we come to cryofixation. And so this was a conference in a long. Everything is old, as you see. All my slides, my references I'm having are very old. So this was a meeting of cryo experts, among others, were there Hans Mohr, Jacques Dubochet, Helmut Sitte, and so forth. All these people who you can easily call were the pioneers in cryofixation and also cryosample preparation. So preparation steps for cryofixation is cryofixation. Let's say samples are at growing or at ambient temperature or even at higher temperature. You fix them with liquid nitrogen. Then we do a dehydration starting at about minus 90 degree. Chemical fixation starts or happens between minus 70, minus 30, and of course higher temperatures. And embedding can be done in the range of minus 50. I will come back to this in maybe the few slides later. Just to show you, if you have no means for doing a good cryofixation, if you get this type of samples, then please use chemical fixation. It will be much better. So this is also maybe to show you how ice damaged samples look like, because very often you see in publication samples that were cryofixed, freeze substituted, and then people say they were amorphous because we used high pressure freezer. This is not true. Look at the samples very carefully. Very often you see segregation pattern more in this range, like here. This probably not. This is really extreme, but in similar to this. So it's very difficult, in my opinion, especially also to properly fix cell culture cells. It's much easier to cryofix tissue cells. It seems that the same cell, let's say what we looked at, liver in liver tissue, so a liver cell in liver tissue is usually much better preserved than a liver cell as a cell culture cell. Now, what's happening during a freezing? You remove temperature, and then you get to a certain point, which is the actually freezing point, and then ice grows, and ice crystal ice crystal formation produces energy. So the temperature goes back to 0 degree. Here we see the different possible freezing points. So usually it's around minus 2 degree in normal circumstances. And this is the energy that comes off during the crystallization, which brings the temperature back so that the sample goes to the thawing temperature of water. And only if all the water molecules are turned into ice, then the cooling continues. And this is exactly what we must avoid if we want to use cryofixation to give us a better structure of our cells. And for this, we need to be so fast that the water molecules don't have the time to reorganize and to find each other to form an ice crystal. And then if we achieve that, then we talk about amorphous or vitrified water. So there were many different possibilities like plunge freezing. And I think I reduced my slides to only the plunge freezing, because plunge freezing is now probably the most used cryofixation method for structural biology. And the first, let's say, sophisticated plunger with an environmental chamber around the sample was developed by Ishitalmon. And he was a chemist, or is a chemist, who wanted to have a chemically well-defined atmosphere around his sample before it was cryo-fixed. And then Peter Frederick then used this idea, or maybe he had the same idea at the same time. I forgot to ask him to develop what we now know as Vitrobot from FEI. So where you can keep your sample in high humidity atmosphere so that the protein don't dry before they are cryo-fixed. And of course, what you see over here, you can then define structures, atomic structures of your molecules. And that's why this is probably the most used technique nowadays, because everybody wants to have a cryo-electron microscope and wants to look at proteins and define molecular structure of proteins, probably much more than people who want to look at the cell biology. So if you want to, of course, this method is only possible for thin layers, let's say in a few nanometer, maybe 1, 2, 3 micrometer thick. Much thicker samples, they don't freeze well, and you will find ice crystals. And if you want to go to thicker samples, which we need for cell biology, then you would need to change the physical properties of water, like adding cryoprotectant, or then using high pressure method, which was invented by Hans Mohr at the ETH in Zurich. And he is using this strange phase diagram that water has, namely that if you increase the pressure, not the temperature, if you increase the pressure, then the freezing point is lowered. And it's at about 2000 bar, it's lowered to minus 21 degree. So that means the dangerous temperature between freezing point and the recrystallization point, if you have already amorphous water, this stretch becomes smaller, which means that even if you have slower fixation speed, you still can get amorphous or well, let's better say well frozen samples. Okay, so this is one of, or let's say this would be my favorite machine. This is from Volvent Engineering, who uses the same principle as the first high pressure freezer from Baltec was constructed. So we have a pressure cylinder and a piston that translates the pressure from 200 bar to 2000 bar. And then this pressure piston pushes liquid nitrogen through a pipe onto the sample and sprays it onto the sample. And with high pressure freezing, probably we can get, depends what you're looking at, between 100, 200 micrometers. Sometimes I have the feeling cell culture cells, maybe 50 micrometer, 100 micrometer tissue, you can easier get the 200 micrometers. And depending if you have samples that itself have some kind of cryoprotectant, for instance, by having a higher concentration of sugars and so on, then you could even go thicker. But in general, I think 100 to 200 micrometers should be okay. And of course, this is then the method of choice if you want to look at the tissue. And then After the sample has been frozen for, let's say, thin sectioning applications, or if you want to do immunolabeling on thin sections and so on, you would go through a freeze substitution process where you exchange the ice for an organic solvent. And many different solvents were tried, like acetone, diethyl ether, methanol, ethanol, tetrahydrofuran. But after all, I think most of the people went back of using acetone, which was one of the first used freeze substituent. And in contrast to chemical fixation, we start then with a dehydration, meaning that at minus 90 or a little bit below, you cannot go much below, because I think acetone solidifies at minus 95 degrees, so you have to be somewhere in that range. Then the organic solvent will remove your ice, will dissolve the water molecules and replace with the solvent. If you add fixatives, and that's probably the nice thing also for freeze substitution fixation, the fixative will then, with the solvent, be distributed through all the cell, but not yet fixed. So you won't have a fixing gradient as you have at room temperature, where you start fixing, for instance, at the cell membrane, and then slowly the fixative goes further in and also hampers itself to move inwards by the fixation. So you can, at room temperature, you usually cannot fix samples which are much bigger than a couple of micrometer in a decent way. So anyhow, coming back to here, what has also been shown or described in a paper, osmium fixation probably starts in at minus 70 degree. And what is actually interesting is that at that temperature, osmium probably looks like I have shown you before. Then it's in oxidation state of 6, so from 8, the osmium tetroxide, to the osmium that binds to 1 or to the 2 CC double bonds, would be in oxidation state 6. And if you look at room temperature, when the sample gets black, osmium is at oxidation state of 3 and 4, and cannot look like it's the molecule that would be bridging. And maybe I can add it already now. Preliminary experiments, which we never really worked out, showed that if you would use osmium fixation during freeze substitution, you will not have such a dramatic effect on immunolabeling as if you have if you're using it at room temperature. With this, I conclude for myself, of course it's a hypothesis because it has not been proven yet, that osmium fixes at minus 70 degree, but at room temperature, as I said, it is proteolytic. So anyhow, then you go higher up in temperature. Here I showed our very first stepwise raising the temperature as we were using in Martin Müller's lab, but of course you can also take a linear or sigmoid curve, whatever you like. Then at higher temperature, the glutaraldehyde starts to fix. And finally, you end at the temperature you want to embed. So here I showed a few metacrylates. So the HM23, you could actually go down to about minus 80, I think, even. I never really, I used it a couple of times, but I didn't really get friendly with this substance. I preferred K11M, which you can use at minus 6 down to minus 60, or HM20. I use both of them at minus 50. K4M I don't like, but it was one of the first. And also LR Gold you can use at about -20. And if you want to use LR White or EPON, which you need to polymerize, usually you need to polymerize at whatever 60 degree. LR White you can change the initiator. You can use the Lovicryl initiator also to use it at lower temperature. But most of the time I use it as the formula as you buy it. So then I start the first incubation of 1/3 of resin with 2/3 of acetone. I start at the end temperature of my substitution, so let's say at -40 degree. And then I bring them after 1 or 2 hours or so, I bring them to room temperature, and then I continue with the other steps, 2/3 and 100% at room temperature until I finally put it in the oven to polymerize at 60. Okay, then here is one example which shows you that you can keep lipids. Actually, this picture, oh, I have almost no time anymore, but I think I'm close to the end. So this picture actually defined my career because I had a long discussion with RF Clay about lipids. He said thin section and lipid, that doesn't work. And I said to him, I don't know much about lipids, but we can look at lecithin vesicles if you use Freeze-Substitution. And so he gave me his famous lipid with me when I was in his lab, and I prepared this sample for him, and he was completely convinced that Freeze-Substitution is really a technique with which you can study lipids as well. And then embedding, there is, we have epoxies, we have metacrylates, and we have sucrose ice embedding. With the infiltration, we also might result in denaturing and dissolution because these are very potent solvents as monomers. And polymerization may bind to epitopes, so might reduce your immunolabeling possibility. So let me speed up a little bit. The epoxies, they can covalently bind to your proteins. Good sectioning quality, smooth surface, and epoxy can also be used as a fixative. We had this example here, and this is also a picture I very much like because it fights dogma which you might have in your labs. So usually, you are not allowed to use, as we said already, osmium, but you're also not allowed to use epoxy, EPON, and so on if you want to do immunolabeling. So my colleague Lianchun Wang, he was looking at pituitary glands and at neurotransmitters. Here is one of them. And he did high pressure freezing of pituitary glands, Freeze-Substitution in glutaraldehyde containing methanol embedding in HM20. So what you would do if you want to do immunolabeling, but he couldn't get label. And then I looked at the molecule and said, this is not enough amino groups for getting a nice fixation, nice fixation of this molecule. And I suggested him to use EPON. And now you see these holes are filled with a dark structure. And also here we can nicely see the gold label. So whenever in your lab something doesn't work, don't state to the dogmas, try to bridge those dogmas. Probably it will help you to solve your problem. And here you can see how the epoxies can fix with hydroxyl, NH2, and sulfhydryl groups. Lovicryls or metacrylates in general, they do not covalently react with your molecules, but they create a mesh where they are trapped in. So we have an initiator that falls into pieces under either UV radiation, which you use for the lovicryls, or under heat, which you use for LR white, making a radical, and this radical reaction is then propagating, making the network. Here we see surfaces, different surfaces of EPON is quite smooth, HM20 gets a little rougher, and K4M is even rougher. And with this, you can easily imagine if you want to do immunolabeling, if you have a protein somewhere here, then it gives better access to your antibody than if you have it here in this smooth EPON, because antibodies will not penetrate your resin. They will only see what is exposed to the surface. And with this, we are coming to. I will be fast. We come to the sucrose ice embedding, which was developed by Tokias. That means the sample is infiltrated with high concentration of sucrose in water and then frozen. You can freeze it in liquid nitrogen or even in the chamber of your cryo-ultramicrodome, because there is so much sugar in the water that you won't have freezing anymore. Only solidification of your mixture. So the advantage is the macromolecules stay in water environment. You need to have, of course, a cryo-ultramicrodome. You will have a very rough surface, as we see in another slide. And with this, we have excellent labeling properties. So here is the slide. These were Tokiyasu cryosections, re-embedded in EPON and then cross-sectioned. And with this, you can see that you get holes, you get breaks off. Again, you have another hole, or you have, thanks to the reduced fixation, usually you don't take as strong fixation or as high concentration of your fixatives for Tokias, for immunolabeling, than you would use for structural preservation. So if the fixation is weak, you can get penetration of your antibodies on gold. And yeah, this is one of the last slides when we tried to combine the advantage of cryo-fixation and of the Tokiyasu technique. So this was cryo-fixation, freeze-substitution, and freeze-substitution fixation, and then rehydration, sucrose impregnation, and then again down to the Tokiyasu. And these were the results of that, where you see that the morphology of this cell is quite nice. And with this now, I come really to the last slide with Ipeni Ferebitan, which is the local language of Okinawa, for saying thank you very much.